I am happy to be here today to share some of the research that we are conducting here at the Ocean Pollution Research Unit. I am just an honorary research associate, but really empowered me to come here and to get inspired by all of you, the professors that we have. And today, I want to just share one of the latest research in terms of the new kind of emerging pollutant that we are observing in the ocean, and also what is next for the orcas, right? So before that, I want to introduce my collaborator, my co-author. Kaya was an undergraduate student here at UBC, and she did basically the work, the analysis of data for 2 papers that were already published. And our adjunct professor, Dr. Stephen Rafferty from the BC Ministry of Agriculture, Food and Fisheries from the Animal Health Center, he basically provides access to a unique bank of tissues of killer whales that were collected in the last 10 years. Of course, we have Paul Cottrell also. He is the manager of the Marine Mammal Response at the EFO. He's the one that's in charge to go to this remote area to collect, to put in place all the equipment, basically, to collect the sample and do the necropsies with Stephen. It is a lot of work. During the summertime, they have to go to all this remote area to try to rescue and try to do some necropsies and take samples. And Dana Price is also a student here at UBC. She's doing microplastic research in sea otters, our BC sea otters, the keystone species. And Zainab Sobeda Danpour, she's from Iran, and she came all the way to help us to understand the impact of hydrocarbon pollution. She's an expert in oil spill in hydrocarbon. Basically, she's now a postdoc at SFU. With that in mind, I want to initiate this talk. Yeah, this is always the colleague will go with it.
Try it now. Should work.
So, as Dave mentioned, I came from Ecuador, right? It's a very small country in South America here. And this is also ancestral land, so what we call the ancestral Manteño or Huancavilca culture that used to be thousands of years ago. Now there is basically nothing about that, but we have also orcas over there, and they are beautiful. They are very iconic animals. This is a photo from a friend that was in the Galapagos many years ago, Benito, and he sent that photo and made my day just to see how beautiful are these killer whales. I know Josh is interested in doing some research with killer whales in the Galapagos. But I am not here. I have migrated from Ecuador all the way here to try to help to contribute a little bit to the issue of ocean pollution in the oceans. And what is important about is that this is basically a powerful spiritual wing also for the local people, for the First Nation communities. It really bridges the science in terms of the communities, the traditional communities, for sustainable ocean. And the orca is part of what we call the spiritual ecology, because it is part of the human connection that really empower the strength, the compassion, the union, the family union. And it's important species to conserve because it's part of the ancestral traditional knowledge of our waters. But the environment is changing for the orcas. And because of the human activities, the anthropogenic impacts, they are basically facing many threats here. And why the canary in the coal mine? The canary in the coal mine is not really a new concept or metaphor. It was used like 100 years ago during the mining times. What happened is at that time, they used this little canary, the Atlantic species of canary, and they put it as an alert system to go inside the mine to see if there was any toxic gases from carbon monoxide, for example. You can imagine if the canary came alive, it was okay to enter, so the miners were fine. But the canary came probably in bad shape or dead, it wasn't safe to go inside. So this is a new concept that we are applying today for all these apex predators in the global ocean. Of course, the orca is one of these apex predators, like the polar bear in the Arctic, like the rigid seal in other part of the planet. But this is important when we want to talk about what are our canaries in the coal mine, right here. For us, the orca has become this canary in the coal mine that tell us how healthy or how polluted are our ecosystems here. So this is a picture from a good colleague of mine, Dr. Lance Barranella. That photo was taking further north in British Columbia, close to an area where we see this smelter here, and these are some workers. It really tells us that the pollution footprint is a testament of our own existence in this water, right? Because we are observing that the habitat is being impaired in terms of all these emissions, all these pollutants that have been basically emitted from more than 40, 30 years, 50 years here. And on the demand date of the effort, for example, this is important because the idea is that chemical and biological pollution do not prevent the recovery of resident killer in our waters. But still we see that there is this group of what we call persistent organic pollutants, right? That have been like the legacy, the classic pollutant that has been assessed for many years, but they are still in the environment because they are persistent, they are toxic, they are bioaccumulative, right? That's what we call the PBT category, persistent, toxic, and bioaccumulative. But there are new ones coming, new contaminants are already in the environment, right? And that's are the, which are not that new because they are related with oil spill, what we call the hydrocarbon or polycyclic aromatic hydrocarbon. It's a kind of infamous one because if you remember what happened in 1989, anyone, Alaska, Southeast Alaska, 1989, in April 1981, the Exxon Valdez oil spill. What was the most catastrophic? It killed a lot of orcas, a lot of sea otters. Even today, you can see that kind of legacy in the sediments or the gravel on the beaches. You go deep inside and you can see the slick, the raw oil still there. So, question bigger here as to whether our orcas were exposed to this contaminant. Again, the classic box that we already know that they are there, dioxin, furans, the concentration are declining for dioxin, furan, for PCBs. We are observing kind of declining in some species, but in some other, they are not really going down because of this biochemical cycle that we see in the ocean. DDT is more prevalent, for example, in the southern areas, like in California and water offshore of the South Bay in California, and also in tropical areas, because it was used to control malaria. It was the cheapest and most effective. I think today, the World Health Organization allowed to use DDT by climate change. It's exacerbated by all this movement of biovectors, and all DDT is still being used. If we want to think about orcas, we need to think about what we call the multimedia environment. The multimedia environment for our orcas is basically the air, the atmosphere, the water, the sediments, of course, the industrial, the anthropogenic activities, right? Of course, they are also part of this environment. They are the biotic compartments, right? They are the recipients, basically, all this pathway of exposure that we can see here. Of course, we have new contaminants in addition to this one that we already know that are there. So now triclosan, norphenol, alpha hexabromocyclododecan, and also the famous forever chemical, the PFAS, which are making the news today. They are everywhere, basically, right? So also hydrocarbons, right? But it's not only the habitat of the orcas, it's also the habitat of the main diet, the Chinook salmon, right? And Chinook salmon basically make up 70 up to 90% of the diet of the southern resident killer. And also the salmon in across all the stages, either the stream life stage, the oceanic life stage, are exposed also to several contaminants coming from different activities like agriculture, the application of pesticides in many areas. As a result, of course, DDT in the past and more recently, you can observe this new kind of current use pesticide, the atrazine, hydrotroids that can be kind of endocrine disruptors for some species, right? We will talk more about that. Also organophosphates, they don't know why accumulate, but they are very neurotoxic, means that affect the nervous system, right? Right now, the smoking gun, the 6-ppd quinone, this is a kind of chemical, it's a toxic chemical, it's being used in tires of cars. It's an anti-ozone, but this anti-ozone used in many cars basically is leaching in the environment through what we call tires particles coming from car. They are transporting this chemical to the environment at such point that we know today that there is a particular species of salmon, coho salmon, that is very sensitive. Just a very small concentration of this chemical can kill 50% of a population. There's a 1 part per billion, basically, right? So, Chinook salmon is also facing these chemical threats. And not just here in the environment, but remember that they can travel all the way to the North Pacific, right? This is the data from Donna Coulomb. She did a nice study in 2009. Basically, she used the eco data to show the routes of migration, the movements of Chinook salmon, for example, from different locations. She showed that they can go all the way here, right, where they can be exposed to what we call the global pollution. That pollution that's coming from trans-Pacific air pollution, like from other part of the world, right? Coming from Asia, from other areas. And it's here where they also can pick up a little bit of that global pollution and can deliver back to the areas where they were born, right? So here, either in the local environment, but in the outside environment, they are exposed to these pollutants. And of course, solar residents are basically heavily reliant on Chinook salmon for survival. Basically, Chinook salmon is the prime item that deliver all this calorie, all this reserve to healthy reproduction, to healthy survival. If they don't have that much of this, there is a huge problem, right? A male orca, like 5,000 kilograms or 5 tons, eat about 5% of the total body. That means like 250 kilograms. If we assume that each salmon can be 50 kilograms, how many salmon are those? It's like 5 salmon per day, basically, they need to ingest to try to survive, right? And this is marine mammal toxicology 101. In 1 minute, I want to show you the basic thing that you need to understand in terms of the marine mammal toxicology, right? So imagine that basically we start with this kind of black tide. I call it black tide because the toxic spill, for example, can be any kind of spill like oil. Hopefully we don't have this here. And what happened with the road exposure? Basically, there are 3 road exposure when you are exposed to a chemical or contaminant. One of them is the contact, dermal contact. Another one is the inhalation via air. Remember, killer whale, marine mammal, breathe air like us. They are air-breathing organisms. Another one is the direct injection via the diet, right? The diet is the main pathway also exposure to all these bioaccumulative compounds. But also there are another kind of pollutant that we cannot see. That's the invisible pollution. That is silent pollution. Remember Rachel Carson, Silent Spring, when DDT was used, we cannot see really DDT, but it's there. So that kind of pollutant also can come to the food web, right? And can accumulate in the prey and also go all the way to the organs. So number 2 are basically all these pollutants that can also accumulate via the food web, right? That can cause, of course, some impairment in the reproduction, can affect the immune system, and also affect the growth of the population growth at some level. And the third one is that the same chemical that we have here, 1 and 2, also can affect the prey, right? The invertebrate. So we see all this kind of pathway of exposure in the food web that basically conspire against the survival of species and also can affect the health. And when we talk about the health, there are many endpoints, many mechanisms of toxic action or mode of toxic action. One of them is immunotoxicity, when the immune system is weakened by this, with this contaminant and makes species more susceptible to disease. The other one are the endocrine disruptor. Endocrine means the endocrine system, the system that regulates the hormones, all these special chemical principles that help in the reproduction of organisms, like thyroid hormone, one of the most important hormones for development. And reproduction, of course, can be impaired, and that's a problem. Right? You have a high pregnancy fatality or high mortality of calves, it's because something is wrong, not only in terms of the food that is available, but also the chemical that are conspiring against the survival of the species. One of the first studies were done by in the mid-1990s when the first sample were collected via biopsy sampling and was published by Dr. Ross. Dr. Ross, a colleague of mine. So basically, at that time, that's more than almost 24 years, the publication by the samples went from the 1990s. He showed that our killer whale was one of the most contaminated marine mammals in the world because of these high levels of PCB. And that was, for example, evident for the big or transient killer whale, which can reach like about 350 milligrams per kilogram. That's part per million. That's pretty high in an organism. And also the southern resident that reached about 200 milligrams per kilogram. That's also high. He found different, like male, for example, can accumulate more contaminant than female. And that's because male doesn't have a mechanism to offload or to deliver this contaminant through babies, but females can do, right? So what happens is that females have this capacity to transfer contaminant via the placenta and also via the nursing, via milk. So the levels in the female will go down over time, but the firstborn in that population is the one that's doing, basically it's a toxic spill, it's a cocktail, not only the lipid, the milk that it receives, but all these contaminants that the mother is transferring via the placenta when it's a fetus or during the lactation. So there is lactational transfer and also placentary transfer. And that's one of the things that we observe also in the new contaminants. There are other sentinels that also can tell us something about that. What species it is? Anyone can tell me what species is here? How do you see Phoca vitulina, right? One of the most abundant marine mammals here in the sea. So it is a kind of sentinel. There has been a systematic monitoring of this species here in the state of Georgia. So some research shown from Ross also that basically PCBs have declined from the 1980s until the last decade, right? But other ones, of course, were increasing during the 1990s. Remember the famous flame retardant, the one that used to make a code for this kind of furniture and also for computer. When regulation came, of course, they get a steady state of they should be declining right now because there are no more production of the formulation for this specific PVDE. But that's the case for a specific group of contaminant, which is a specific kind of marine mammal that can that easily to capture and also to take biopsy from there. That's more complex when you are working with killer ones, right? But the truth is that we have a problem here in the state of Georgia in terms of the critical habitat, the home range of killer whales. When I was still before come here, we did a work with GFO with several colleagues to try to see whether the sediments that are here are protective of killer whales. And guess what? They are not, because this sediment, as you can see, both for Chinook salmon, when we did the modeling, basically the concentration, the sediment predicted forward to the concentration in Chinook salmon, one of the areas close to in this area, close to the habitat, critical habitat orca. Basically, most of these concentration were exceeding at that time the quality guideline that we have from the Canadian Council of Ministry of Environment, right? So it means that even for Chinook salmon was not protected, but also the same scenario was found for the orcas, right? Basically, in most of the critical habitat, in sort of the home range for the orca, all these concentrations that were calculated forward based on the sediment concentration were not protected. You know why? Because these guidelines were formulated to protect benthic invertebrates. So we were losing all the trophic them in this food. We forgot the top predator in the system. So this work that we did, basically, we calculate through food web bioaccumulation modeling using what we call the biota sediment accumulation factor, a concentration that should be protective of at least for the 95% population, right? So today, 10 years later, the federal government is going to publish the first document, which is the environmental quality guideline to protect apex predator in Canada, based on 3 species: the southern residents from PC, the ringed seal from the Arctic, and the belugas. So these 3 species were selected as a representative from the 3 oceans of Canada to come with this environmental quality guideline that was formulated for the sediment from the water and also a tissue residue guideline that can be protected. So that will be the regulation for the next years. And I will start with the first chapter of this. And this work basically, this is another thing I want to show, that we were observing declining, but this was a work that we did here. We did here with Rashid, with William and Andres. We tried to see whether climate change was exacerbating this contaminant in food webs. So we were wondering whether, the increase in ocean warming, acidification was conspiring with the contaminant to see an increase in the food web or the orcas, for example. We observed, for example, for the diet for salmon, that under the high emission scenario, also for the low emission scenario compared to the baseline, it means 0 emission. Basically, you see like exacerbation of this contaminant over time after 2050, right? So you can see that climate change is somehow exacerbate what we call bioaccumulation or biomagnification of pollutant. But this is just for the Chinook salmon, right? The concentration in this case, we were most focused in PCBs and also on mercury. Here in the y-axis, you see this concentration predicted with the model versus time in the x-axis. For our class, of course, we were observing a decline because PCB were phased out, but still, even in the decrease compared to the reference baseline, you can see that still you see this exacerbation or when we do this under the high emission scenario, right? So still you can see that the pathway of exposure and somehow can be changed by the changes in temperature and also by the changes in the food web composition that can really exacerbate the accumulation of this pollutant in the food web. And that was part of the history at that time. Today, in collaboration with the federal government, I am part of what we call the toxic working group, the technical group working group for on contaminants for the southern resident killer group, right? So there were like several meetings since before the pandemic. Since 2018, we were doing several meetings with many scientists from the academia, from NGO, from Environment Climate Change Canada, from DFO, to try to prioritize what pollutant we should really continue monitoring or assessing to try to come with some kind of environmental quality guidelines to protect the species. So we have tier 1, tier 2, and tier 3, and we can see here the contaminants that were selected. And this was part of the rationale that we took as a kind of primer to initiate this work with the bank of tissues that we have access to. So, and I want to just go quickly because we don't have to time. But one of the contaminants that we are focusing this research at is the PFAS, so forever chemical. These are very much like using everything in a fire extinguisher, even in the pans that we used to make the pancake, right? They fixed it out already, this, but they were here, right? And also another kind of application. And what is complicated is there's a long chain of carbon with fluoride, and that's what makes this contaminant very sticky. And that's why they use as a flame retardant or water repellent, right? And they can be neurotoxic, they can impair the immune system, right? So they are used in many things. Here in Canada, of course, there is a regulation that prohibits already, not all of them, but at least P4 and P4, which are one of the most common PFAS that we observe in marine mammals, and also what they call the long chain PFCAs. And this is a new one that is coming right now, and it's really being observed in marine mammals. This contaminant wasn't 5 years ago observed, but right now it's becoming more prevalent. PFOS are declining, but it seems that when you want to control one, there is another one coming, a new one that is showing, right? And another kind of contaminant are the what we call the alkyl phenols, and these are used in the detergents and also in the famous toxic toilet paper that also is very problematic because this is a potent endocrine disruptor. This is the one that causes feminization in invertebrates. It's a problematic one, and there is a strong evidence. We are concerned because the levels of non-phenol that we were going to see later were in high concentration in this organ. So this is a potent endocrine disruptor, and it's very hydrophilic and also hydrophobic. The other one are these flame retardant. It's called the hexabromocyclododecan. It's a kind of flame retardant. We suppose that it was a substitute for the PDDs that we saw at the beginning with the harbor seal, but they are already kind of toxic and they can be problematic also for marine mammals. They are used basically in all furniture as a flame retardant. That's the goal, right? It have benefit like PFAS, forever chemicals have benefit, but they are basically a double-edged sword, right? They can cause problems in the organisms. And this one, the triclosan and methyltriclosan, basically were used, most of us use this, right, in toothpaste to clean our teeth, but they are phased out already here in Canada as well. But they are still in the environment because they are also persistent. They have a higher bioaccumulative potential. So that was one of the issues with triclosan. And right now, it should be phased out. They shouldn't be in any product that we use as for antiseptics, for example, or detergents, right? Because they are potent endocrine disruptors. The last group is what we call the hydrocarbons, the polycyclic aromatic hydrocarbons. And basically, we know the history of these pollutants in many areas of the world. And there are several sources, right? They are what we call the petrogenic. They are coming from crude oil spills. The pyrogenic are coming from the burning of biomass, the plant, the carbon, the coal, for example, coming from wildfire. All the biomass that is getting burned basically can produce what we call the pyrogenic one, and there are other coming from organisms and transformation process in the sediments. Of course, here is some example for petrogenic, and this is, for example, pyrogenic one. And there are several groups. There is probably several of these contaminants, but usually we focus in like 12 or 17 that are like the one that indicate contamination by pHs. These are alkanes, different from the polycyclic aromatic hydrocarbon. Aromatic means rings. Basically, this benzene ring, right? That's what makes really problematic to break down in the environment. These one are alkanes. These are also prevalent in oil spill. But these alkanes, like CH4, like methane, like propane, like are very prevalent dominant in oil spill, but they are more easier to be break down because bacteria can eat like a pigment. So they come in carbon, they break down basically that. So this is the problem, basically, this one here. Even when benzopyrene is metabolized in an organism like orca humans, it can produce a byproduct, a metabolite, that is the one associated or the one that causes basically what we call mutagenic effect or adducts, DNA adducts, right? Like the benzopyrene. Basically, this oxidation during the enzymatic metabolism produce this byproduct, is very the one that causes carcinogenesis or is carcinogenic in organisms. So basically the idea, as I mentioned at the beginning, was to contribute with this new kind of reserve on contaminant, emerging contaminant, and a new POPs like the PFAS, and also the hydrocarbon, because we didn't know nothing about whether hydrocarbon were in the orbits, right? And to investigate maternal transfer and identify potential pollution sources. We don't have time to go to this, but the idea with Stephen, because he's a pathologist, he's a veterinarian pathologist, it was interesting to see whether these chemicals were associated with disease in the organs. At the end of the day, it's not just to do all this analysis to collect, but at the end of the day, the idea under the mandate of the FO Environment Climate Change Canada is that this data can be provided as information. So decision making can be put in place to really control, to really hamper the emission of these pollutants. So to do that, basically, we have access to many kinds of strands, not many, with 6 other residents and 6 big killer wells, right, that were found stranded. Basically, most of them in the west coast of Vancouver Island here. Some of them in Georgia, and just one further north in this area, around close to further north, close to Haida. So basically, all these samples were collected from the liver and the skeletal muscle. Were sent to a lab, a special lab that we have here in Victoria and Sydney, one of the best labs to analyze all this data. And we assess the effect of confounding factors to see whether the girth, the length, or the state of the composition of the animal was affecting the presence of this contaminant. Of course, one of these well, was Luna. Luna was one of the stranded animals that were observing Nutka sound where she was victim of a proper interaction, right? So we got sample of Luna. He was an orphan, but he used to be around humans. He used to like to interact with humans in boats and other areas, but also he used to be hanging out around other remote areas. So Luna is a special indicator also in this research. And this is just the table of all the information for all the samples for the period of time, basically, and the different age categories and the sex of the animals that were basically analyzed. As I mentioned, we assessed whether there was some influence of the confounding factors, right? Like for example, the body condition index, the personal lipids, the age, and basically we control for that kind of potential factors. The only one was this contaminant that was observed very, very small amount. It was negligible, basically the concentration of this kind of alkyl phenol. So the concentration was not significant. So we didn't worry about that. For pHs of hydrocarbon, the same history, basically there wasn't any influence of age for the total contaminant load or other kind of parameters. So no influence of this confounding factor. But remember that always this kind of underlying disease, the status of the health of the animal always will influence also our interpretation, our data in the discussion. It's important to discuss that in a paper, right? And this is the research that Lilia did as undergraduate research student here with the help of Paul and Stephen. Basically, she came as part of the summer program. So a student that are part of this program, she approached me and she wanted to do something. She didn't know what to do. And it happened, I was in conversation with Stephen when he published the paper in pathologies of pillar well here. And she was very, very empowered by that kind of paper. And we started research. It took like 2.5 years to send samples to analyze the data because it was unique data to be shown, right? And Luna here, again, here was part of this research. So all these contaminants were analyzed. Basically, we observe here that the most prevalent were the PFAS in this kind of brown color, and also the alkyl phenols, right? Were the one that were the highest concentration, 52% for PFAS and 48% for the alkyl phenol. That were the dominant contaminant, the emerging contaminant observed here. And when we break down by contaminant class, again, we observe that there are particular ones like the 4-nonylphenol, which is prevalent in the detergent and also in sewage. In sewage, and also in toilet paper, and other kind of contaminant like the 7,3-FTCA, this contaminant, there's a new PFAS that was observed here, right? More prevalent in big pillar wells, while the 4-nonylphenol was more dominant for the southern resident pillar well, yeah? This is the concentration on these are the samples. Luna exhibited a high concentration for this contaminant, for 4-nonylphenol, right? Remember that she was interacting a lot with human environments. Another one, when we break down the PFAS, also in a more deep analysis, again, we can see all the pattern of contaminant. Again, 7,3-FTCA is one of the most prevalent, and followed by PFOSA and also by PFOS, which is the color here, the blue, like that blue with this light green PFOSA, right? So there are the more prevalent forever chemicals in this kind of samples from organisms. When we make comparison, we of course observe some differences in when we analyze specifically the calves, from southern resident and the calves of these, we see some interesting patterns, right? So we see more prevalence of the 4-nonylphenol, which is more observed in urban environment because of the sewage, all the emission coming from Roundup. While for the more remote mobile, here basically we observe this kind of 7,3-FTCA because this is a more kind of new precursor that can be observed, for example, in the atmosphere or emitted in remote area when people are using, for example, what we call aqua film forming foams using fire extinguisher, for example, right? Like wildfires. And this is the maternal transfer ratio. And we can see that basically some of these contaminants are transferred, readily transferred from J32 mother to J32 fetus, right? So high concentration basically show in the comparison of the concentration in J32 versus J32 mother here in the y-axis, right, for this group of contaminants. So maternal transfer does occur for this group of contaminants, making basically most of them. The ratio MTR equal 1 is maternal, the maternal transfer ratio, and 1 means that it's equal distributed, equal partitioning. So you are a whole, it means that the fetus is receiving more. We can also see this in terms of the comparison between in a regression line, in which in the y-axis we have the J32 fetus and the J32 mother in the x-axis, and basically we show here this basically the highest low, right, indicating maternal transfer for many of these contaminants. And the dashed line here is the 101 again, in which basically the same concentration mother and fetus, right? So you can see that these contaminants are transferred, and this is the case, for example, for the one that was more dominant, the 4-nonylphenol, basically a potent endocrine disruptor. You can see the structure of this, the chemical structure of this contaminant. And keep in mind that because this is important for placentary transpassing the cell, the membrane organism. But there are others that are transferred in low amounts, right? Like these people, you see the structure, it's a kind of complex structure, and that means that the molecular side, the kind of chemical composition made more difficult to transfer the membrane. So it's not transferred that much compared to nonylphenol, right? So this is one of the implications when you are looking at the molecular weight, the size, and the number of atoms that you have in in these compounds. And this one was transferred the lowest. It was very complex, these rings to be moved from mother to the fetal, but still concentration are kind of low for the transfer here. And when we compare this to the LOCW in the x-axis, this is a metric for bioaccumulation. This is the partitioning between octane or water partition coefficient. It's a metric that used in bioaccumulation science. With the maternal transfer ratio in the y-axis, you can see again this kind of nice curve in which you can see the trends for the contaminant, right? Some of them are transferred with higher amount, while others in lower amount. And the LOCW indicate the potential of this contaminant to bioaccumulate. The highest the KOW, the highest the potential to bioaccumulate in tissues of organisms, right? But remember that there is the highest here are found between 5.5 to 6.6. After 6.6, which go down because the molecules are more complex, more difficult to be transferred. Remember that it's because of this structure of the ring, it's more problematic to be transferred from the placentary members. The implications are important because we know that there are 50% mortality of calves here in southern resident, and the pregnancy failure is almost 70%, so that's high. And this is not just because of contaminant, it's because also of food, daily food. Climate change might be also conspiring. There are many other factors, interspecific competition, right? The prey play an important role here. This is just what a kind of trends that we want to see whether basically there was a kind of trend over time for total PFAS, so forever chemical, and we see that there are some ups and downs, and that might be related to regulation for some PFAS, but for some other that are coming new PFAS, maybe they are basically influencing this trend, right? In terms of the PFAS in our environment. We don't have much time for that, but basically for hydrocarbons, which are basically kind of smoking on because they were observed in our orcas. We also observed maternal transfer for these three first ones, which are basically derivatives, pHs coming from parental compounds, not just the parental, the byproduct, which is more persistent and toxic, basically was the more prevalent to the maternal transfer. And also we see all these pHs hydrocarbons that were observed in high amounts, and the more prevalent were basically phenanthrene, anthracene, which are more related to petrogenic contamination. The same approach we did for the southern residents and the big skiller well to compare the calves, and we see also differences in fingerprinting, different fingerprinting in the concentration of this hydrocarbon. When we calculate the maternal transfer ratio, also we can observe basically that there is a high efficiency for some of these contaminants here, like citrifluorine, benzothiophenone, naphthalene, which are coming from petrogenic or pyrogenic sources here. The same history is repeated here, except for this trend and for the LOCW for some pHs, because of the size of these molecules are more difficult to be transferred from the mother to the fetus, right? But still, we observe that some of them are readily transferred, right? Like naphthalene, which is a petrogenic pHs, while others are transferred in lower amount when we do the comparison between the fetus to the mother and the concentration of maternal transfer ratio versus the LOCW. Some more complex molecules like benzo and anthracene are more difficult to be transferred. That's why we see this kind of decline for some of them, but some others are readily transferred from mother to the fetus. And I want to finish, if time allowed, with the origin, the sources, the petrogenic sourcing for this. Basically, we do some ratio between low molecular weight and high molecular weight hydrocarbon to see whether they are coming from petroleum or oil spill or from wildfire or biomass burning, right? That's what was the goal. And we use a specific also isomer ratio to do this kind of research. We observed, of course, that for some of these individuals, we see that basically low molecular weight are the dominant one, right? For pH, while the high molecular weight are less abundant. Most of the low molecular weight are related to petroleum sources, oil, basically, while these ones are more coming from emission like combustion from burning of biomass, the plant biomass burning of vegetation that has been burned or wildfires, right? So, and this is a dot cross plotting just to show the same by using a specific isomers. Here in the x-axis, you have the fluorine ratio for fluorine plus pyrene, which is a very common hydrocarbon, to see basically the trends. And Luna basically was important to elucidate the petrogenic versus pyrogenic because she was between pyrogenic, petrogenic. She was exhibiting sources coming from burning of organic matter like pyrogenic, but also at the same time petrogenic. Remember that she was roaming around human environment, marine, and also remote area. So this particular individual tells us also a lot about how individual species can be exposed to different kinds of sources, while most of the southern residents that you see here with ratio below 0.4 basically indicate petrogenic, a petrogenic means petroleum, right? While the southern residents were more exposed to this pyrogenic, pyro fire coming from flames. And we corroborate this with the principal component analysis. Again, we have a cluster only indicating here for the southern resident versus this big scalar well. So that also the feeding shape and the PCA explaining 46% of the variation, high loads for the southern resident, and the PC2 only about 30%. So that is important also when you want to further corroborate your data to go deep in analysis, like using multivariate analysis to understand better, right? How this is the driving force explaining the presence of this contaminant in the organism. And these are the conclusions I will be happy to share, but basically it's what I was explaining, and there is more to do. Our orcas are contaminated, that's the bottom line. BC, we have a problem, so we need to hamper emission. We really need to change our way in how we consume fossil fuels also. So that's basically the take-home message. Maternal transfer, of course, did happen in our thank you to the students that were working with this research, and our collaborators and our supporting agencies here in DC. Thank you.